yet another first in V1 is that in V1 you find the first cells which respond selectively to direction of motion. So that's a property known as direction selectivity. And again, I stress that's only in primates, so humans and monkeys. In some other animals, you do find direction selective neurons in the retina, but as far as we know, not in primates. And as you might imagine, these cells are believed to form the initial basis of motion perception, which then feeds on to other brain areas for later processing. I'm going to show you another video from Hubel and Wiesel showing their recording of a direction selective neuron in cat visual cortex. So this is a neuron which Hubel and Wiesel recorded, and as you can see they've labelled it as a direction selective cortical cell. So once again, you remember that these little crackles you're hearing are the action potentials fired by the neuron. The recording has been connected to a loudspeaker so that you can hear the neuron in this way to make it easier for the experimenters to see how the cell's responding. And they're moving this bar across and they're going to try and find out what drives the cell best. So they've marked off with that little box the region where they think the cell is most responsive. And now you can see, hopefully although the image quality isn't good, that they've marked on an arrow indicating what they reckon is the preferred direction of this cell. So I don't know if you find it convincing, but they reckon that they're getting more spikes when they move the bar in this direction than when it moves back again. Now you might criticise them, I think, for a confound. I have the impression that they're moving it more slowly in what they think is the preferred direction and more rapidly in the other direction. So you could argue, is this cell really sensitive to direction or speed? And for exactly that reason, in a modern experiment, this would all be done by a computer, and the computer would make sure that the speed was exactly the same in both cases. But here Hubel and Wiesel are just exploring, just trying to get a sense of what this neuron is sensitive to. It's not a rigorous experiment. So there you could see them measuring out, mapping out the full range of orientations in both directions. And this represents how they recorded their results. So this plot on the left is from a Hubel and Wiesel paper. And so they've got the bar of different orientations moving across the receptive field, and they're showing the spikes recorded in one direction and then in the other direction moving back again. So for this orientation, the neuron isn't firing any spikes for either direction, but say for this, its preferred orientation, it's firing lots of spikes when the bar's moved in this direction and very few when it's moved back in the other direction. So this neuron is tuned both to orientation, as you can see comparing the different rows, but also to direction, as you can see by comparing the left and right parts of this plot. On the right is a similar way of representing that information from a more modern paper, and they're plotting orientation 360 degrees around the clock. So you can see with the arrow, this is a grating that's horizontal and moving upwards, and then 180 degrees away represents a grating that's horizontal but moving downwards. And you get these two peaks in this full orientation direction tuning curve. So the cell most likes gratings that are 45 degrees moving upwards, but you do get a secondary peak for cells that are at that same orientation moving in the other direction. So this cell is clearly orientation tuned, as you can see, but it's also direction tuned because it's responding about twice as much to this direction as to that direction. Now, how does a cell end up being sensitive to direction? One way is if its receptive field, or simply the on and off regions within it, shift over time. So let's think how that might happen. Here I've shown the receptive field of a cell. I've drawn this one with a big circular on region shown in red and then an off region shown in blue. And the idea is that after this cell is activated by receiving visual input, this off region actually moves across the receptive field. So I've shown it at time 1 on the left, and then by time 5 it's all the way over on the right. So what this means is that a cell with this type of receptive field would respond best to a dark bar moving from left to right across the visual field. Okay, so at time 1, this dark bar's over the off region, so we get spikes. At time 2, the bar's moved a bit because it's moving, but so has the off region. So the dark bar is still over the off region, we still get spikes. Same at time 3, and time 4, and time 5. So this cell is really strongly activated for quite a long time as the bar moves across its visual field, and of course its receptive field. And of course it has to be tuned to the contrast polarity of the bar, so the dark bar matching this off region, and also the orientation, and also, as you can see, the speed of the bar. So this is an example where this cell is beautifully matched to the properties of the bar moving across it. Now this cell would not respond so much to exactly the same bar moving from right to left. So let's think about this. So now at time 1, the dark bar is on the right of the receptive field, and so the cell's not active because it's got a dark bar over part of its on region. And I want to note a possible confusion. This bar is still moving left to right across the slide, but hopefully you can see that it's moving from right to left across the receptive field of the neuron. So at time 2, it's moved a little bit left in the receptive field, still over the off region, still no spikes. Now at time 3, we do get some spikes because the bar briefly coincides with this moving off region. But at time 4, no spikes, and at time 5, no spikes. So a cell with a receptive field like this ends up giving lots of spikes for a bar moving from left to right, but very few for a bar moving from right to left, so it's direction selective. Now the existence of these kind of time-varying receptive fields also gives an insight into a phenomenon called apparent motion. So I'm sure you're all familiar with this kind of idea that if you have a bar that actually jumps, flashes, it appears at one location and then briefly at another location, even though there's no actual movement across the visual field here, maybe you have a perception, you almost feel like you can see the bar moving in between those two points, even though, as I say, there's no motion on the slide. Now, if you have a sufficiently large jump, then the motion percept eventually weakens and you just perceive it as a flashing bar here and a flashing bar there without any sense that there was motion across the space in between. And what we think might be going on there is that you get apparent motion when the flash stimuli are close enough together in space and time that they both fall within the receptive field of the same direction-tuned neuron, and so they stimulate it. So here's our neuron again. Originally, in the example I showed you, there was a bar moving smoothly from left to right across it, and this cell was optimally stimulated. In an apparent motion situation, you get a bar here at time 2, let's say, and then a bar over here at a later time. The bar has not moved across the space in between, but you can see it's still going to activate this cell. Not optimally, not as much as a smooth bar that's hitting this off region at all times, but it's still going to give some spikes in this cell, and we think that's what gives you this sense that there was some kind of motion there in that image. If the jump were bigger, then the bar would appear here in the receptive field, but then when it next appears it would be outside the receptive field, so over in this green region here, and so the cell would only be very briefly activated, and importantly cells tuned to leftward motion would be activated just as much as to rightward motion, and so you wouldn't get this imbalance between the two that gives you your sense of motion perception. And so we think that's the neural basis of this phenomenon of apparent motion. which is quite a useful thing for us because, of course, it's the basis of all cinema and video and television, which don't contain any actual movement of anything across the screen. They just contain briefly flashed images showing objects in slightly different positions.